When bacteria have multiple different sugars to use as a carbon source, it will selectively use glucose always first because E. coli, like for example, grows much faster on glucose. And to make sure that they don't waste a bunch of energy creating enzymes that they don't really need for sugars they don't plan on breaking down, they use a mechanism called catabolite repression to control which carbon sources they use.
When cells of E. coli are grown in a medium that contains glucose, the synthesis of enzymes needed for the breakdown of other carbon sources, such as lactose or like maltose, is repressed, even if those other carbon sources are present. Thus, the presence of a favored carbon source represses the induction of pathways that catalyze other carbon sources. Catabolite repression is sometimes called the glucose effect because glucose was the first substance shown to cause this type of response. But catabolite repression is not always linked to glucose. The key point is that the favored substrate is a better carbon energy source than other available carbon sources. Thus, catabolite repression ensures that the organism uses the best carbon and energy sources first. Why is catabolite repression called global control? In E. coli and other organisms for which glucose is the best energy source, catabolite repression prevents expression of most other catabolic operons as long as glucose is present. So dozens of catabolic operons are affected, including those for lactose, maltose, and a host of other sugars. In addition, genes for the synthesis of flagella are controlled by catabolite repression because if bacteria have a good carbon source available, there's no need to swim around in a search of nutrients. One consequence of catabolite repression is that it may lead to two exponential growth phases, a situation called dioxic growth. If two usable energy sources are available, the cells grow first on the better energy source. Growth stops when the better source is depleted, but then following a lag period, it resumes on the other energy source. Dioxic growth illustrated in this figure below. The cells grow more rapidly on glucose than on lactose, although glucose and lactose are both excellent energy sources for E. coli. Glucose is superior and growth is faster.
Cyclic AMP is an important molecule in like metabolic control, and it's often called a regulatory nucleotide because it's made from a nucleic acid precursor, and it's synthesized from ATP by using an enzyme called adenylate cyclase, and the synthesis of cyclic AMP is inhibited by glucose, the presence of glucose, and the presence of glucose stimulates cyclic AMP to get shot out of the cell. And when the cell receives glucose, the cyclic AMP levels fall, and then because of this, when the levels fall, CRP can't bind DNA. And then RNA polymerase can't bind to the promoters of the operons that is being controlled by this. So in a way, like catabolite repression is, it's kind of it's indirectly a result of when like the presence of a more like usable energy source. Catabolite repression uses an activator protein that acts as a positive control. And the activator protein is called cyclic AMP receptor protein. And this like kind of relates to lac operon and like the whole process of catabolite repression because for lac genes to be described, there has like two conditions have to be met. The level of cyclic AMP has to be high enough for the CRP protein to bind to the binding site, and lactose or another like inducer that's like present in, needs to not block transcription by binding to the operator, and the operator acts as a negative control. And then when all these conditions are met, then the cell will like kind of let itself know that there's no glucose, and lactose is the thing that it should start metabolizing. And then once that happens, then transcription of the operon starts.